and the metabolism of amino acids. Okay, so first of all, let me, before starting up with the discussion, I just want to tell you what do you mean by these amino acids. Can anybody define what are amino acids? These amino acids have a zwitterion structure. I'll just explain it to you. Then we will start up with the discussion. Okay. These amino acids are those compounds which can behave as an acid as well as a base, or they have a zwitterion structure. And on the basis of this zwitterion structure only, we can precipitate these proteins. And at the pH, a point at which the proteins are precipitated, that is known as the isoelectric point. Right. So today we will be discussing 40 questions on the amino acids and their metabolism. Okay. Let's see question number 1. Non-essential amino acid group among these amino acids is. Right. So first of all, you can just unmute your mic and tell me. Non-essential amino acids. Okay. So as per the nutritional requirement, these amino acids are actually of three main types. So can you tell me which will be the answer? The answer will be acidic amino acids. Okay, so whenever we have to, differentiate between essential amino acids, non-essential and semi-essential, the basic criteria is this, that as per nutritional requirement. Essential amino acids are the ones which cannot be synthesized in our body. Synthesized in our body. Right? And the mnemonic to remember these essential amino acids is TV till 8 p.m. Okay? If you have any problem, any confusion, you can just unmute your mic and you can say your problem. Okay? Instead of chatting, instead of writing the problems in the chat box, you can just unmute your mic and ask me. That would be better. Is my screen visible to all of you, first of all?
Visible.
It's visible, na? So if you have any problem, beta, you can just unmute your mic and ask me. Because this is for the first time I am using this app, so I have to be a little bit sure of this ki how this app works, right? Well, let me just give you my brief introduction. I am Dr. Pooja and I will be discussing the biochemistry portion with you. Okay, so if you have any kind of problem, don't hesitate to ask me. So have you little bit idea about the nutritional requirement of the amino acids? Essential are the ones which cannot be synthesized in our body. So they have to be supplied from outside diet. So there is a mnemonic for this. And this is a very, very important question which is generally asked in the PG exam. So T stands for threonine, V stands for valine, tryptophan, isoleucine, leucine, lysine, phenylalanine, and methionine. Right. These are the essential amino acids. Then comes semi-essential. Semi-essential are two: arginine and histidine. Now, out of these semi-essential, this histidine sometimes come under essential amino acids, and arginine is conditionally essential. Okay, and after this, rest of the amino acids will be what? They will be non-essential, which can be synthesized in our body. Now, let's look at the question. The question says non-essential amino acid group among these amino acids is. So, acidic amino acids are aspartic acid and glutamic acid. So, as I listed, the essential and semi-essential aspartic acid and glutamic acid does not come under that category, so it will be non-essential. If we talk about branch chain amino acids, in branch chain amino acids, leucine is there, valine is there, and isoleucine is there. So these are essential ones. If we talk about basic amino acids, we can say lysine is there, arginine is there, and histidine is there. So again, we saw they are essential. And last but not the least, aromatic amino acids, it comprises of phenylalanine. Right? So the answer to this would be A. And you can just see the essential ones. Histidine is there, isoleucine is there, leucine, methionine, phenylalanine, threonine, tryptophan, valine, and lysine. And conditionally non-essential are arginine and rest. So now you got to know what is the mnemonic for essential amino acids. You got to know
TV till
8 p.m.
TV till 8 p.m. Yes.
Yeah, 8 you have to, 8 is just a, just to give a significance to that sentence. Otherwise, TV till PM. That's it. Okay. And this is a very important question, in fact. Then moving on to question number 2, which is a 21st amino acid. Now see, if we talk about amino acids, there are 20 standard amino acids. But there are certain exceptions to it. 21st amino acid, does anybody know the answer to this question? 21st amino acid is selenocysteine. Right. There is one more, 22nd. 22nd is pyrrolysine. Selenocysteine is coded by UGA and pyrrolysine is coded by UAG. The only irony with these two amino acids is that they are coded by nonsense codons. Okay, and all of you must be having an idea about what are the nonsense codons. Nonsense codons are the ones which do not have a corresponding anticodon, and that is why we refer them to as 21st and 22nd. So the answer to this would be selenocysteine. Right. And a very important point about this is that it is selenocysteine, means it has selenium with it, plus it has cysteine with it. And always remember one thing about selenium, this is a trace element. And selenium, the major function of this is it works as an antioxidant. Antioxidant means it prevents the oxidation, it prevents the proliferation of the free radicals. And it is referred to as 21st amino acid. And this selenocysteine is synthesized in our body by a very important enzyme, which is simple selenocysteine synthetase. Right. And this is used as a coenzyme in so many important reactions. This is a coenzyme for glutathione peroxidase, thioredoxin reductase, right? So the answer to this question is selenocysteine. As you can see, 21st and 22nd is UAG. This is pyrrolysine and this is selenocysteine. As such, pyrrolysine ke functions, we are not clear of the functions, but yes, selenocysteine is very important. It is a component of enzyme glutathione peroxidase. Similarly, it is required for thioredoxin reductase. Right? As you can see, selenocysteine, the 21st amino acid, it is found at the active site of so many enzymes like glutathione peroxidase, glycine reductase, thioredoxin reductase. And see, cysteine combines with selenium, as you can see here, and it forms selenocysteine. And the enzyme I told you, it is selenocysteine synthetase. Right? Then, okay, the 21st amino acid present in these enzymes, just now I discussed glutathione peroxidase, deiodinase, glycine reductase. One more is missing, I'll write it here, thioredoxin reductase. Right? Now, as you can see, it has a structure similar to that of cysteine, but it has selenium with it. And it is coded by UGA, which is a stop codon or a non-sense codon. Right? So this is 21st amino acid, which is selenocysteine. Right? Then next question, amino acid present in thioredoxin reductase. Now you tell me what will be the answer to this? The amino acid present in thioredoxin reductase, it would be, just now I discussed. Better you can unmute your mic and tell me the answer.
Selenocysteine.
Yes, selenocysteine. So the answer is selenocysteine. Right? As you can see, this is the function of this. Do you know what is the role of glutathione peroxidase in our body? Anyone? What is the role of glutathione in our body? Yes? Let me see, what have you written? Antioxidant, correct, very good. This is the function, yes. Okay, I'll just explain this reaction to you because this is very, very important. I'll just show this reaction. One sec. My screen is visible now also, na?
Yes, ma'am.
Okay. Actually, this is sounding to me like Google Meet. In Google Meet also the condition is the same. Then I'll search for some solution here. Now see what happens is, I'll just show you this reaction because this is very, very important. Glutathione peroxidase. This glutathione is a tripeptide which whose main role is as an antioxidant. Okay, so this glutathione occurs in two forms. This is oxidized form and this is the reduced form of glutathione. Right. Now, if we have to convert this oxidized to reduced form, we require NADPH. This NADPH will be converted into NADP positive and it will convert it into this reduced form. And always remember, when glutathione is in its reduced form, it will lead to the degradation of hydrogen peroxide like this. And selenium is a component of this. Now, selenium is a trace mineral. Trace mineral means it is required in a very minute quantity, but still it is important for the body. Why it is important? Because it is a component of glutathione peroxidase. And if we say that selenium is absent or NADPH is absent, this glutathione will not be in its reduced form. And when glutathione is not in its reduced form, this H2O2 will be converted into free radicals. And when our body starts forming free radicals, that's the worst stage of our body. So these free radicals they have to be prevented from proliferation. That is why antioxidants play a very, very important role. Right? So here you can say this is a very simple, that is what is shown here, reductase. This is thioredoxin. Thioredoxin means sulfhydryl group. And this pentose phosphate cycle, this is actually a shunt which generates this NADPH. Right? So the answer to this question will be simple, selenocysteine. Then moving on to the next question, glucogenic amino acid is. Now again, whenever we talk about this amino acid, I'll just show you the structure of this because this is a very, very important question. And always remember, every year a question comes on this. We have the structure of amino acid like this: NH3 positive, COO negative, and R. So this structure is similar to all the 20 standard amino acids. So how we categorize their catabolism or metabolism as per nitrogen skeleton, right? And as per carbon skeleton. So whenever we talk about carbon skeleton, we divide it into three classes or categories. One is glucogenic, right? Second is glucogenic as well as ketogenic. And third is ketogenic. Again, there is a mnemonic for this. See, always remember, I know biochemistry students, they have that idea that biochemistry is the toughest subject. But if you study biochemistry from clinical point of view, it is the most interesting subject. And most of the things, most of the terminology is hidden in these only. See, genic means genesis, means formation. So glucogenic amino acids are those which give rise to glucose or its precursors. Ketogenic means genic, genesis again. So they will give rise to some keto group. And glucogenic and ketogenic means they can function as, they can give rise to glucose as well as they can give rise to keto groups. Again, there is a mnemonic for this. Always remember, leucine is a potent ketogenic amino acid, right? Then PITT. This is a mnemonic for those amino acids which are glucogenic as well as ketogenic. P stands for phenylalanine. I stands for isoleucine. T stands for tryptophan, and this T stands for threonine. So they are glucogenic as well as ketogenic. Except these, rest are, and one lysine is also there which can act as, predominantly it is ketogenic, but to some extent it shows glucogenicity also. Apart from these, all the rest are glucogenic. And always remember, alanine is considered to be the most important glucogenic amino acid. So now if we look at the options, question is glucogenic amino acid is. Ab now see, leucine is ketogenic, right? If we talk about lysine, lysine is ketogenic and glucogenic both. Phenylalanine, glucogenic as well as ketogenic. The option left is valine. So valine is a strictly glucogenic amino acid, right? So the answer would be valine. You got my point? Leucine and lysine. Leucine is ketogenic. Lysine is mainly ketogenic, slightly glucogenic. But phenylalanine is both glucogenic as well as ketogenic. So the left one is valine, which is only glucogenic. Right? Ab this category is there. You can remember this by this, or you can remember the way I have told you. So it's up to you. Because these are the questions which are actually asked in the exam. And if you compare what I wrote and what is here, it's almost the same. Always remember, leucine is the most ketogenic, because I remember 4 or 5 years back there was a question for PG entrance only, that which is the most ketogenic amino acid. So their options were leucine, lysine both. So students got confused between that which one to take. So always remember, whenever there is an option between leucine and lysine, you have to correct leucine. Always remember this. It is strictly ketogenic in action. Right? Then again, question based to that. Both glucogenic and ketogenic amino acids are all except. Now you give me the answer to this. Just see what I have told you just now. Question is, both glucogenic and ketogenic amino acids are all except. No one.
Leucine.
Because leucine is a potent ketogenic amino acid. Tryptophan, phenylalanine are both. Similarly, tyrosine. So the answer will be leucine. Correct. Then indole ring is present in. Now see, the format of this question is little bit incorrect. I will just correct it in a sense that here it should be, see, tyrosinase is the name of the enzyme. Okay. Phenylalanine is correct, and this is tyrosine. So here it should be tryptophan. Right? So always remember, tryptophan has a indole ring. Now, do you know what you have an idea of what is an indole ring? Indole ring has this type of structure. This is like this. CH2, CH, NH3 positive, COO negative. This is the benzene ring and this is the indole ring. So this indole ring is a characteristic feature of only and only tryptophan. Always remember this. And there is a specific test for this indole ring, which is Hopkin Colles test. The reason why I am telling you all this is because these are the questions which have been frequently asked in PG exam. I am into this field giving coaching for PG for the past 10-15 years. And I have seen the pattern of questions, ki what kind of questions are asked in the exam. And these are the ones which are asked. So indole ring is a characteristic feature of only and only tryptophan, and it gives a positive Hopkin-Colles test. Right? So the answer will be simple tryptophan. Just correct it, it should be tryptophan. And this is tyrosine. I'll write it here, tryptophan. Then see, the structure is also here. You can see phenylalanine contains a phenyl ring and tryptophan has a indole. Indole. This is the indole ring. This. Right? Then D form of the amino acid is derived from. Now you know what is the concept behind D and L form. What is the concept between D and L form? Anyone? This is my first class with you. Okay. And if you have any problem related to any of the questions, please don't hesitate to ask me. The purpose of, it is a privilege that you people are still getting, in this, getting an online coaching in this type of pandemic, it itself is a blessing. So whatever problem you have related to biochem, you can ask me anytime. Today our class got delayed just because of, because this is for the first time I was using this app. So automatically next time there won't be any problem. So I hope whatever we are discussing, you are understanding the things.
Yes, ma'am.
Okay, Khyati, are you understanding? Now, can you tell me indole ring is present where? In which amino acid indole ring is present?
Tryptophan.
Tryptophan. Okay, Khyati is not speaking anything. Whosoever wants to speak, they can just unmute their mic and they can ask. Okay. So, we were talking about D and L amino acids. Now, see what happens is whenever we talk about these, always remember in our body, if we say L amino acids are present. Why L amino acids are present? Because of the prevalence of L amino acid oxidase. Right? And if we talk about D amino acid, first of all, you should know what is an L amino acid, what is a D amino acid. Now, see, this is the structure of an amino acid. I'll write it again, NH2, COOH, and R. Always remember, if NH2 group is on the right hand side, it is D amino acid. And if NH2 group is on the left hand side, it is L amino acid. And in our body, most of the amino acids are present in L form. Right? What the question says, question is about D form of amino acids. So I spoke about the the amino acids which are there in the body. So here it will be from the external source, because L-amino acids degradation takes place in the liver, breakdown is also from the muscle, and they are synthesized also in the muscle. But we are talking about D form, where NH2 group is on the right hand side. So it will be from external source. So the answer would be C, right? As you can see from the explanation also, that L-amino acids represent all the amino acids found in proteins during translation, or you can say in the body itself. Whereas D-amino acids are found in some proteins. So D-amino acids we always get from the external source. Nowadays, so much of research is carried on these D-amino acids. And some people have come to this conclusion also in this schizophrenia and all. These D-amino acids are responsible. But otherwise, overall, if we see, the L-amino acids are the one which undergo active transport and they are utilized by the body. Then amide group containing amino acid is. Amide group is actually the NH2 group. Right. Up, can anybody tell me? See, again there is a little bit problem in the question. Glutamate and glutamic acid are the same. Glutamic acid, you can say the acidic form is glutamate. But here amide group means glutamine, because this glutamic acid is converted into glutamate, glutamine, and aspartic acid is converted into asparginine. So here the answer will be glutamine, right? As you can see here, answer will be glutamine. And if you see the structure also, this is the structure. Aspartic acid is converted into asparginine and glutamic acid is converted into glutamine, right? Then guanidium group. Always remember, guanidium group is associated with arginine. There are three. Let me see the structure is there. Yes, this is the guanidium group, as you can see here. Actually, lysine, arginine, and histidine. These are the three basic amino acids. Arginine is characterized by the guanidium group, phenylalanine by benzene group, and tyrosine by phenyl group, right? So here you can see this is guanido group, and this is alpha carbon atom, alpha, beta, gamma, delta. So at the delta position, guanido group is present. So here the answer will be arginine. Then amino acid which is not stable in alpha helix. Always remember, alpha helix is the secondary conformation of a protein, and the only amino acid which is never present is proline. This is a very, very important question, because proline has this type of structure. Wherever proline is present, it is going to destabilize the helix, right? And the answer will be simple A, right? As you can see, proline disrupts alpha helix. Glycine also, but why might glycine not participate? Glycine doesn't participate in the secondary structure, but wherever proline is present, it will disrupt the helix, right? The next, glutathione. Now, glutathione is a tripeptide. It is formed of, this is a mnemonic for remembering it, GCG, which stands for glycine, cysteine, and glutamic acid. So glutathione is a tripeptide formed of glycine, cysteine, and glutamic acid. And it is a very, very important antioxidant. Just now we studied that. Right? So the answer is simple, a tripeptide. Answer is C. And this is the structure of a tripeptide. You can see GCG. Glycine related to cysteine, related to glutamic acid. Then amino acid with a double chiral carbon atom. Now double chiral carbon atom, always remember, again there is chiral carbon atom is a asymmetric carbon atom. Asymmetric carbon atom means all the four substituents attached to it are different. So this you have to learn, that I will just write it down. This you have to learn, that glycine does not have a chiral carbon atom, right? Because it is the simplest, amino acid. And threonine and isoleucine. Nobody is going to ask you the structures, but you should know. Threonine and isoleucine have two chiral carbon atoms. And if we look at the question, double, tyrosine, no. Threonine, yes. Tryptophan, no. Phenylalanine, no. So the answer is threonine. Just remember this. Answer is this. So threonine and isoleucine, these are the only two amino acids which have two chiral carbon atoms. This is the position. This is the structure also, because this is the structure of threonine. If I have to draw, this is the structure. COO negative, CH, CH3. You can see one chiral carbon atom and second carbon atom. Threonine is there, so it will be OH here. One sec. This will be OH. CH2 and CH. So this is a chiral carbon atom. This is another chiral carbon atom. And that is why the answer will be this. As you can see, there are two chiral carbon atoms in threonine. First is alpha carbon atom bonded with the amino group, and second is located in the side chain. This is the side chain, and this is the alpha carbon atom. Then negatively charged amino acids are, always remember, the acidic ones are the negatively charged ones. These are positively charged. These are positively charged. These have hydroxyl group in them. So the answer is this. D. Right? This is the structure. You can see negative charge. They have an extra carboxylic group. These are the polar amino acids. Then this is a very important question. Substitution of which one of the following amino acids in place of alanine would increase the absorbance of protein at 280 nanometers. Now always remember, only the, I'll just write it here, aromatic amino acids absorb at UV range, which is 280. So अब यहाँ पे you have to find out which one is a aromatic amino acid. So leucine, arginine, tryptophan, proline. Tryptophan is a aromatic amino acid which absorbs at 280 nanometers. So the answer would be tryptophan. Always remember this. This is again a question which was asked 5 or 6 years back. Right? 280 nanometers. This is the absorption of the aromatic amino acids. Right? Same, it is like this, to roughly here. So you can see aromatic amino acids are at UV range. Then creatine is made up of all except. Now see alanine. जैसे glutathione is made up of GCG, creatine is made up of GAM. Glycine, arginine and methionine. Right? So, is made up of all except it is arginine, methionine, alanine. So, the answer would be B. And this is a very important reaction which takes place in two organs, liver and kidney. First reaction takes place in the kidney where arginine combines with glycine. Second reaction takes place in the liver where methionine combines with this forming creatine. This creatine, when it loses a molecule of water, it is converted into creatinine. And creatinine we see in urine, and it is a very, very important example of a renal function test. Right? Then amino acid required for the formation of thyroxine. It is tyrosine. I'll show you one slide, one sec, it is there. Answer will be B. Now here what you have to remember is phenylalanine forms tyrosine in the body. This tyrosine is required for the synthesis of T3, T4. It is required for the synthesis of catecholamines. And it is required for the synthesis of melanin. So you have to remember this, that tyrosine is a precursor of T3, T4, triiodothyronine and tetraiodothyronine catecholamines, epinephrine and norepinephrine, and melanin, which is the skin pigment. Right? This is a very important question. This is most of the times this has been asked in the exam. Next, disulfide bond is seen between. Always remember, disulfide bond is seen between sulfur containing amino acids. Containing amino acids. So if you look at this, the sulfur containing amino acids are only cysteine and cysteine. The spelling is wrong. It should be cysteine. This cysteine has this type of structure, SH. And this is the amino acid. So CH2SH. Simple hydrogen bond is given out and it forms a disulfide bond. So a disulfide bond is present between cysteine and cysteine. Actually, cysteine and cysteine ही होगा. This is cysteine basically, so it can be correct also. Between two molecules of cysteine, cysteine will be produced. Right? This is transsulfuration. This should not come in this reaction actually. This is disulfide bond. As you can see, SH, hydrogen goes out and SS is produced. Then sulfur containing amino acid metabolism yields. Now always remember, this is a very important question. There are two sulfur containing amino acids, methionine and cysteine. Methionine is required for the synthesis of cysteine, and it requires B6, B9 and B12. So we can say that pyridoxine, folic acid, B12 all are required for sulfur containing amino acid metabolism. So here the answer will be D. Pyridoxine is B6, folic acid is B9 and B12 is B12. As you can see, this methionine forms cysteine, and here this requires B6, right? And this methionine is forming homocysteine. Here B12 and B9 are required. This is B9. So you can see in that reaction B6 was there, and here B9 and B12 are there. Then coenzyme required in the formation of glutamate. It is niacin, and the active form of niacin is NADP, and the enzyme is glutamate dehydrogenase. As you can see here, tryptophan, niacin, glutamine to glutamate. Niacin is actually vitamin B3. Is niacin. Then which process involves formation of non-essential amino acid from keto acid? Now see, non-essential amino acid, which means it can be non-essential is glutamic acid. From keto acid, this process is transamination, which means transfer of a amino group from an amino acid to a keto acid to form a new amino acid and a new keto acid. These are new and these are old. So this is a non-essential amino acid. So the answer is transamination. B. Transfer of an amino group from an amino acid to a keto acid. As you can see here, this is what is transamination. Then this is a very simple question. Amino acid absorption is by, always remember, it is absorbed by active transport. Always remember this. Against the concentration gradient. This is a straightforward question. Answer is C. Right? And you can see the picture also. Active transport is energy mediated. Right? Then branch chain amino aciduria. Now see, MSUD type 1A is due to the mutation of this. I'll tell you, it is E1 alpha. Always remember. If we talk about E1 beta, it is 2. And this MSUD is actually due to the, the deficient enzyme is actually, this is the metabolism of branch chain amino acids. When this alpha keto acid decarboxylase is absent, right, there is accumulation of these in the urine, and the urine starts giving the smell of burnt sugar. So the answer is simple, E1, right? In this, lipoyl transacetylase enzyme is deficient, and in here decarboxylase is absent. Decarboxylase. This decarboxylase consists of TPP. This decarboxylase is without TPP. And here it is transacetylase. As you can see, maple syrup urine disease caused by mutation in E1 alpha subunit is referred to as type 1A, E1 beta type 1B, and that caused by defect in E2 is type 2. And this is what is simple, caused by deficiency of ketodehydrogenase complex or decarboxylase complex. And the person who is suffering from MSUD, there is mental retardation is there, and there is excretion of these branch chain amino acids, and urine gives the smell of burnt sugar, and that is why it is known as maple syrup urine disease, right? So till now it is clear. I hope till now the description is clear. So we have discussed today the explanation of 23 questions, and rest of the questions we will be discussing tomorrow, because the time is going up and you have to attend the other class also. So is it clear to all of you? Any confusion up till now? If there is any confusion, you can ask me now also. Is it clear, Tabish and Khyati? So next of the 14 questions we will take up tomorrow. So shall we wind up the class now? Okay. So good night. See you guys tomorrow.